I'm Lieutenant Colonel Craig Myatt, President of the Faculty Senate and Faculty Assembly at the Uniformed Services University. On behalf of the Uniformed Services University President, Dr. Richard Thomas, the university leadership, the Faculty Senate, and Faculty Assembly, I would like to welcome you to the 2021 David Packard Lecture, where we are hosting Dr. Robert Gallo. The Packard Lecture is named in honor of Dr. David Packard, the co-founder of the Hewlett-Packard Company. Dr. Packard served as the Deputy Secretary of Defense from 1969 to 1971. He served also as the second president of the Uniformed Services University from 1976 to 1981. The David Packard Lecture is considered the Uniformed Services University's most prestigious lectureship. The Packard Lecture has been offered annually for over 36 years since 1985. To introduce and welcome the 2021 Packard Lecture speaker, please join me in recognizing Dr. Mark Kortepeter, the Uniformed Services University Vice President of Research. Dr. Kortepeter.
Thank you, Colonel Myatt. And good afternoon, everyone. And I also would like to welcome you to this Packard Lecture. And first, I'd like to thank our Faculty Senate, and particularly Dr. Marjana Pazgier, for their significant efforts in putting this event together. It's been more than a year in the making, so thank you all. I would also like to recognize several distinguished guests joining us online, including Brigadier General Shan Bagby, the commander of Brooke Army Medical Center in San Antonio, Dr. Ada Sue Hinshaw, Dean Emerita of the Daniel K. Inouye Graduate School of Nursing, retired Lieutenant General Tom Travis, Senior Vice President for USU's Southern Region and former Air Force Surgeon General, Dr. Carol Romano, current Dean of the Graduate School of Nursing, Dr. Thomas Schneid, Executive Dean of USU's Postgraduate Dental College, Mr. Tim Rapp, Vice President for Education and Information Technology at USU, and Mr. Bob Thompson, USU's Chief of Staff. So thank you all for being with us. And we're extremely fortunate to have Dr. Gallo today as our speaker this year. So Dr. Gallo is currently the Homer and Martha Gudelsky Distinguished Professor in Medicine and the co-founder and director of the Institute of Human Virology at the University of Maryland School of Medicine. He's also the co-founder and international scientific advisor for the Global Virus Network. Dr. Gallo has a long and distinguished medical, scientific career. He was the first to identify a human retrovirus and the only known human leukemia virus, HTLV, which is one of the few viruses shown to cause a human cancer. In 1976, he and his colleagues discovered interleukin-2, which is a cytokine signaling molecule that regulates the activities of white blood cells and has been used for therapy in some cancers and even HIV. In 1986, he and his group discovered human herpes virus 6, which was later shown to cause the infantile disease known as roseola. And of course, he's recognized internationally for his co-discovery of HIV as the cause of AIDS. Dr. Gallo and his team also pioneered the development of the HIV blood test, which then enabled healthcare personnel to screen for the AIDS virus for the first time, leading to a more rapid diagnosis. This also protected patients receiving blood transfusions, and ultimately this work helped physicians develop HIV therapies. As a biomedical research scientist, he since has spent much of his career working to eliminate AIDS and other chronic viral diseases. On a personal note, his work has had a direct impact on my career. When I was a medical student, I was in the trenches in Newark, New Jersey in the mid-1980s when we were hit with an onslaught of patients with HIV. And we were all afraid to catch this horrible new infection of HIV. So having the blood test was important for our ability to treat our patients and also to understand the risk in the healthcare setting. And it's really been a wonder over the course of my career, over decades, to watch the evolution of HIV, where the patients back in Newark in the 1980s would come in off the street and they would be dead within a week. And now we have the fact that they can live relatively normal lifespans. So Dr. Gallo, thank you for your work, and we are honored to have you with us today and look forward to hearing your thoughts. And now the floor is yours. And you're on mute, sir.
You're good. You're live.
Okay, I hope you can hear me now. No, they don't.
Yes, sir.
Okay, thanks. Well, good afternoon to everyone, and thank you for your very kind and generous introduction, Dr. Corder-Peter. And thank you to the Faculty Senate, thank you to the Faculty Assembly, to President, this year's President, Colonel Myatt. I would even say thank you to President Thomas, who is away at this point in time, and a special thank you to my former colleague, one of your illustrious faculty, Marzena Pazier, who carried out some great molecular biology while she was with us at the Institute of Human Virology in Baltimore. I would like to go through different phases of my early and mid-careers and later career, 3 phases at the National Cancer Institute across the street from you. And in that, I would cover the discovery of these retroviruses. And then I want to tell you about more recent interest, like everyone else, in COVID-19 and its causative agent, the SARS coronavirus number 2. So to begin with, I'd like to tell you that I start at a time when no one else but me could remember, because it's 1965 when I came to the National Cancer Institute. I was then involved as a clinical associate and really new into laboratory research. I had done some as an intern, believe it or not, and as a resident at the University of Chicago, but really nothing like a full-time position in it. And after 6 months on a leukemia ward, I entered full-time research. And my interests at the very start were the biology of blood cells, and especially comparing normal and leukemic cells. But at the same time, I had a developing interest in how did leukemia occur? I had come from the leukemia wards, and I had a sister who passed away with leukemia, so it was very much on my mind that if I did research, it would be along those lines. But I knew I needed extensive background technical experience, biochemistry, and the just developing field of molecular biology. If we go back to that period, there was a healthy respect for infectious diseases causing serious epidemics, possibly, and possibly even an occasional pandemic. Yet, roughly 10 years later, it was forgotten. That is to say, it was forgotten that serious infectious diseases really could occur, could spread, could become epidemic and pandemic. And there were certain lessons that come out of that. So we're now in the 1970s, and that's a period that I'm really starting to do research at NIH. And the statement I have at the start, serious epidemic infectious diseases are over. That's what's said in the industrialized world. Therefore, we can forget about them. You may say, was there evidence for that? Major medical schools, famous ones, terminated departments of microbiology. That should say enough. It was a problem for tropical disease institutes, and mainly only for them. Research money should go anywhere else but. They should focus only on the chronic degenerative diseases. That was a general and strong feeling. Secondly, retroviruses, which we had become interested in, and I'll tell you that story in some detail, do not infect humans at all. And there are many reasons for this. That was emphatically stated. Thirdly, no viruses cause cancer or play any role in cancer, even as a cofactor or secondary role. Evidence for it? Look at the 1974 Cold Spring Harbor Laboratory Conference on the Origins of Human Cancer, edited by James Watson and Jim Cairns. That was a conclusion. All cancer was accidental mutations or some chemical, period. However, by the early 1980s, these biases were shattered. Viruses were then shown to be involved in the cause, maybe the whole cause. In most cases, it was one step in the cause of about 20% of human cancers. Retroviruses were discovered in humans and shown to cause some leukemias. fatal neurological disease, and modest immune abnormalities. This was the first human retrovirus, kindly mentioned already in the introduction, human T-cell leukemia virus, sometimes called lymphotropic virus, number 1, 1980. First, we didn't have the 1, but a year later, we discovered HTLV2, a second human retrovirus that we found in a hairy T-cell leukemia. The first one was involved in a vicious adult T-cell leukemia, both of CD4 type. However, HTLV2 is hardly pathogenic, and for the most part, we can forget about it. And then finally, one of the great pandemics of history appeared, AIDS, caused by still another retrovirus. Well, how did these developments occur? As I mentioned at the start of my talk, my early interest was human blood cells, their growth, their differentiation, normal versus leukemic. And we had to find ways to grow them. To study them, we'd need to grow them. Now, you may think automatically, yeah, you look for growth factors or something, but the concept of growth factors was newly emerging. You remember that Stanley Cohn and Rita Levi-Montalcini got a Nobel Prize for finding epidermal growth factor and nerve growth factor, really much after that time. So the concept of growth factors was new, but conditioned media, that is, if you put cells in a culture flask and you let them sit around, they may release factors, which included growth factors. It was a new concept. I did not invent that. The two individuals I just said were early involved, but also in Israel, Leo Sachs, who discovered colony stimulating factor and was using it to make short-term growth of granulocytes, but not sufficient to do biochemistry or some forms of molecular biology. They were small colonies. We needed cells growing richly in culture fluid. T cells were only just discovered. Hard to believe for many of you in this audience of mainly, well, all of you, younger than me. So, but think of it. T cells were just about being discovered. But soon thereafter, Peter Knoll in Philadelphia discovered that the plant lectin phytohemagglutinin made T cells divide. But they only divided once. And it was firmly thought that an antigenic stimulation of a T cell, the newly discovered lymphocyte, T cell, only could divide once. We didn't know enough not to hunt for growth factors that made cells grow more permanently in culture to be able to use to study. So we continued our quest to find and make blood cells grow. And our success first came with using the conditioned media, the fluid surrounding PHA-stimulated normal human blood lymphocytes that turned out to be T cells. We discovered in 1974 that those cells made the growth factor for granulocytes that Dr. Sachs had discovered, colony stimulating factor. But this was controversial because it seemed illogical that one cell type, a T cell, would control another cell lineage, a granulocyte. In fact, Professor Sachs told me that it was silly. But this was the beginning of a concept of cytokines. One cell regulating another cell, even of another lineage, much like T cells do broadly in our immune response. A few years later, in 1976, we found that the very same conditioned media, that is, once again, the fluid, the media that is surrounding PHA-stimulated lymphocytes, which would be T cells, also released another factor, another protein. We called it T cell growth factor and published it in Science. Today it's called interleukin-2 or IL-2. Some say this is the very first cytokine. It's certainly the first interleukin, one leukocyte impacting another. But simultaneously, we became interested in looking for human retroviruses. Why? Because of our interest in the origin of human leukemia. And in animals, retroviruses were shown to cause leukemia in many species. I should give credit at this point to Dr. Robert Ting, T-I-N-G, a Chinese-American scientist who influenced me while we played tennis together. He not only influenced me about Chinese cuisine, but also about tennis, but finally also about virology. He had worked in the laboratory of two Nobel Prize winning actually people who came from Italy to the United States. Salvatore Luria at MIT, and then Renato Del Becco in California at Caltech. He worked with both of them and convinced me that I should be following closely these kind of viruses, not yet called retroviruses, but called leukemia viruses, or sometimes RNA tumor viruses, because their genetic information was in the form of RNA. And he was convincing me to follow the career of a scientist named Howard Temin. I'll come to him in a moment. But looking at the slide, you'll see that we knew that chickens' leukemia was caused by a retrovirus, that mouse leukemias were caused by a retrovirus, cat leukemia, cow and sheep leukemia, and in gibbon apes, Japanese-American Tom Kawakami discovered that chronic myeloid leukemia in nature was caused by a retrovirus, gibbon ape leukemia virus. And then our group found in late 70s that a T-cell leukemia of gibbon apes was caused by a retrovirus. Nonetheless, there was strong resistance to looking even for the possibility of a human retrovirus. Why was that the case? Well, there was a few decades of searching already by many with completely negative results. We thought, well, we need more sensitive techniques. The argument against us was that high replication in known animal models, when you had a leukemia in an animal model, it was high replicating. And on the other hand, we knew that the cow leukemia, that was not true. And there may be many more animals which you don't study because it's too difficult. You only use animal models when they're easy. So we didn't like the argument that it should have been easy to find them. There was little evidence in primates as disease-causing agents until the late 1970s, as I've already referenced. Then a group showed that human serum in the presence of complement could destroy animal retroviruses. Therefore, humans were protected, but not true. We found later that the human serum with complement could not destroy human retroviruses or even several of the primate retroviruses. Finally, and it's hard to believe, but take my word for it, at that time, human research was far less appreciated among basic scientists, so-called basic scientists or laboratory scientists. It was looked down upon as less sophisticated. Of course, that's no longer true. It leads the pack now. And cancer, as so to speak, a catching infectious disease, seemed a very primitive concept. But it was not understood that this could be a slow virus infection, causing disease over a very long period of time, and the person with cancer not really being the one spreading the virus. But things started to look better for the discovery of human retroviruses. Howard Temin, that I already mentioned, the scientist that my friend Dr. Ting said I should be following his career carefully, predicted that retroviruses, again, they weren't called retroviruses yet, but this group of viruses, leukemia viruses, replicated by RNA going to DNA. This was an unheard of concept at the time. It flew in the face of the central dogma of molecular biology, which is that from DNA, you made DNA, or you made a messenger RNA, but you never reversed it, RNA to DNA. Temin thought differently. In chicken system, Rous sarcoma virus, in avian leukemia virus, he saw that there seemed to be a latency period, and it was in the form of DNA that the virus was hanging around. He spent the 60s being thought of as a foolish person, really laughed at. Just like Ludwig Gross in the 50s was laughed at for mouse leukemia virus, which was at that time the first mammalian leukemia virus. But he was greatly abetted by his own discovery of a special enzyme, reverse transcriptase, which we will refer to as RT, confirmed at the same time by David Baltimore. The two of them published together in Nature in independent papers. Reverse transcriptase is a DNA polymerase, that is, an enzyme, ASE, that catalyzes the making of a polymer. The polymer is DNA. So it's a DNA polymerase, but it makes DNA directed by RNA. Really amazing. This provided the real basis for Temin's provirus theory, but this was augmented by terrific molecular genetic studies, chiefly, but far from solely, by Peter Vogt in California, who is still active. Making reverse transcriptase, RT assays, sensitive and distinct from human cellular DNA polymerases took a great part of the 1970s for my colleagues and myself, but also major contributions, again, from Baltimore. This is a number of postdocs might have thought they spent some wasted years at that time. But we now knew how to distinguish this viral DNA polymerase, RT, from all the cellular DNA polymerases that are carried in normal cells. But two things drove us to T cells. One was our capacity to grow human T cells best of all, better than we could granulocytes or other blood cells, by using interleukin-2. And then our isolation of a new strain of gibbon-ape leukemia virus associated with T-cell leukemia. Those in white was the work that led to the discoveries. In orange are the advances that kept us looking. Our approach was to develop methods to grow primary blood cells using growth factors, coupled with finding ultra-sensitive but specific assays, namely the enzyme RT. Reverse transcriptase, RT, provided three major advances. It provided a mechanism for Temin's provirus theory. Second, it became a powerful tool impacting molecular biology in several ways, including a major impact on gene cloning. But for us, number three, it also became a surrogate marker for a retrovirus. We thought it might. It did. It was 1,000 times more sensitive than electron microscopy. It allowed for frequent sampling of cultures for testing for a new virus, especially important for viruses that are released in periodic bursts, which can be the case and usually is with human retroviruses. We focused on T-cell disease, we already said this, and ultimately we discovered and repeatedly isolated the first human retrovirus. We published this in 1980. After characterizing it in some detail before publication and finding it repeatedly in the same individual and antibodies in his family members, as well as in himself, we showed that this was, along with Japanese workers who followed us, the cause of CD4 positive adult T-cell leukemia and of a paralytic central nervous system disease, which was then used to be called tropical spastic paraparesis, now usually referred to as HAM, HTLV1 associated myelopathy. I already mentioned HTLV2, and to forget about it, this is the first electron micrograph of the first human retrovirus shown budding from the membrane of a leukemic T cell at the top, and then the leukemic blast cell below. That's the first human being from which a retrovirus was ever isolated. His initials was CR. He came from Mobile, Alabama. He didn't have an interesting personal history, work history, or anything else, or family history. He was a young man playing basketball and with an occupation. The lesions on his body are not uncommon. They're infiltrates of the skin of leukemic blasts. This disease is often associated with a high blood calcium. The mechanisms are known, and a very aggressive course, not always, but mostly. So this is the beginning. So he repeatedly isolated from him and then from others, which we don't have time to go into now, of course. The epidemiological information is important on HTLV1. The number infected globally is unknown because it's not studied adequately or enough, but it's estimated between 10 and 20 million. In other words, 100% error. Of public health importance where? Not in the United States or in Europe, but in Japan, South America, especially Peru, Colombia, and Brazil. Strangely, Romania, strangely, Iran, Caribbean islands, Africa, and the people from Africa who came to the Americas, a small percent will carry this with them. Melanesia, the Aboriginal Australians, and Indonesia. There are approximately 100,000 infected in the United States. It's fixed tightly in a population, almost like a genetic marker. In other words, it's very poorly infectious. It can vary among neighbors greatly in incidence. For example, 3% or 5% or even more in some Caribbean islands and not present virtually at all in some others. Depends on their ancestors, tribal ancestors who carry the virus. How is it transmitted? Well, it's transmitted by blood, therefore blood transfusions, drug addiction, by sex, and by mother to infant. That should raise some thoughts in your mind. Doesn't that remind you of a future retrovirus about to come, HIV? It causes not one disease, but several. It partly depends on the subtype of the virus. And that kind of reminds me to the medical students that might be able to hear this talk, that we learned in medical school or graduate school or even before. One gene, one protein, no longer true. You can modify a gene sequence, splicing, for example, but there are other ways, and get more than one kind of protein. One protein, one function. No, not really true. One protein, one enzyme, not always. And then you could say one microbe, one disease. When I went to medical school, that was more or less the thinking. Not true. Certainly HTLV1 can cause several very different diseases. Oh, I'm sorry, I went too fast. I can't go back. Oh boy. Oh, I apologize, folks. I'm trying to get back to the slide that I passed by. I'm getting a little help. And one slide, no, is that right? Can you go back again? Oh, okay, now we can go forward. Thanks. I'm okay. So HTLV1, I said, causes T-cell leukemia. And I'd like to be provocative. I think it's good. It raises debate. I believe there's more evidence for the cause of this leukemia by this virus than we have for almost anything. A genetic disease like sickle cell, okay, that's more definitive. But in almost any way you look at it, you can argue causation for this virus. Epidemiologically, the fact that it transforms T cells in vitro, the fact that its provirus put into a mouse as a transgenic will recreate leukemia, the same kind of leukemia, T-cell leukemia. And then, oh, there are other arguments, but this one is as definitive as it gets. The integration pattern. Look on your left, virus is infecting a T cell, CD4 T cell. Notice that cell in the second layer, the provirus is integrated. That's the red within the chromosome. Notice it's in the middle chromosome in one place and that all the descendants, it's in the same place. That's clonal integration, indicating that the original cell giving rise to the tumor was infected, not a later cell, implying that the infection caused the transformation. If the integration pattern was found to be random, it indicates that infection occurred only after transformation. The strategy for HTLV to survive, to me, it depends upon propagation of the integrated DNA provirus, because there's very little viral replication. It's strange but true that infection tends to be requiring the whole cell, the cell and its provirus. When a cell gets into your blood from someone that's infected, it meets your cell and gives rise to activation of your cell. And the provirus, the DNA, the provirus is the DNA containing the genes for HTLV1 are transmitted to the recipient cell, thus propagating the spread. Then under certain circumstances, there can be activation of the integrated provirus. Activation of the provirus and its regulation and steps that lead to the transformation of the cell to a malignant cell, it deeply involves some genes that are special to HTLV1 that you can't find anywhere else, it seems. One is called the TAX gene, TAX, and another is called HBZ. This regulation of gene transcription is done predominantly by this TAX gene and its protein product. And important work has been carried out in this regard by someone at your own medical center, Dr. Joe Jim. So his work is focused on that integration, using it to study gene transcription as a model but also to study the mechanism of how a given gene product, in this case, the tax protein of HTLV1, can lead to profound changes in DNA, chromosomal changes, mutations, all kinds of dysregulation. That is a subject, not of a seminar itself, but the subject of a symposium. But for me, in a broad sense, I want to get back to, I think the survival of this virus depends on the proliferation of the T cell containing the provirus, and that adult T cell leukemia is an occasional accidental consequence. So I had jumped about the mechanism. That's the last part of the slide. I said it first. That was my mistake. Now, these are a list of the known infectious causes of human cancer. But I'd like to point out and use the slide only for one reason, to show you the exceptional oncogenicity of HTLV1. You don't hear about it very much because it's not common in the United States. Take a look at the third column, the lifetime risk of malignancy development. It's about 5 to even more % of the infected persons that'll get leukemia from HTLV1. It's a lot better than lung cancer with cigarettes. It's a lot better than almost any carcinogen we know of in man, and certainly better than any other candidate tumor virus. The next best being the hepatitis viruses. I mean, EBV is very low. So are all the herpes viruses in their ability to cause any kind of cancer, yet you know about them much more. But of course, they're far more prevalent, and EBV is contributing to far more disease that we see in the Americas for sure. But if you're born infected from your mother's milk, usually, it's as high, according to Graham Taylor at Ohio State, as 20% of all infected babies will develop leukemia when they're young adults. This is not anything to sneeze at. I want to tell you that from work of particularly Antron Jassain's group, and he used to be a postdoctoral with us when I was at NCI, and Vefa Franchini, who came as a postdoctoral, but of course is the head of a laboratory or a section at NCI for years now. She used to work with me, but very independent. And work with them and work that they did, really it's their work. They been involved, and a few others, another postdoctoral who's in Switzerland named Koralnik also contributed to this work. But you will see that there are subtypes of HTLV1, like there are of HIV, but nowhere near the variation. And the subtlety of those subtypes, variation one from another, is minimal, but yet they exist. The cosmopolitan type, subtype A, is the dominant, which you see in orange and green in different parts of the world. Subtype B is only in Africa. Subtype C, mainly, if not only, in Australia among the Aboriginals. You'll also see some oblong, or pardon me, rectangular bars that says NHP to H, non-human primate to human transmissions. There is known to be at least 3, a few in Africa and one from Asian monkey to man. These are ancient transmissions from primate to man. It cannot happen in the Americas because the monkeys in America don't harbor an HTLV1 related virus, which we call simian or STLV1. The subtype C in the Aboriginals is important. They're poorly studied, but you'll see why it's important in a moment. By the way, yeah, that emphasizes, I mentioned some of these people before. They are mentioned here now, which is a journal. If we look at this place called Alice Springs, take a look at the number of people infected with HTLV1. In males, it approaches 50%. Overall, it's about 45, 46%, male, female. Enormous number of infected people. We have ideas why, but I don't want to get into it. The interesting thing is they are very frightened of the virus. They have little help for it, but now Australia begins, as we had some meetings there, to become aware of it and provide funding to do seroepidemiology. But excuse me, before you look at this slide, let me tell you what they're dying of is a completely different disease. It's bronchiectasis. They get some kind of immune impairment. No one understands it. No one barely is studying it on Earth. and they're having a fantastic mortality rate. Okay, and they're begging for help. So this has been, well, it's not a forgotten disease, just an unknown disease. I show you this slide. It's one molecular biology slide I wanted to show on HTLV1, and it shows the various genes and splicing mechanisms. But if you look at the bottom part with the orange, you'll see the dip below the line occurs right in that region near the 3' end. That means that the greatest difference in the different types of HTLV1, if you look A versus C, it's in that region. The same if we took the B type, it would be in that region. Thus, something in that region is responsible for HTLV1 causing more leukemia. HTLV2 differs in that region mostly from HTLV1 as well. It is not causing leukemia anywhere near that rate. And HTLV1C, they're never seeing the leukemia, but what they're seeing is bronchiectasis. But in the leukemia-causing one, we also have the high incidence of the paralytic neurological disease. Sorry, it's not clicking and going forward once again. Oh, I'm sorry, folks. We have to give a timeout. It's not working. And I'm getting behind. Okay, yeah, I want to go forward from there. So that region is what I was talking about. Oh, geez, Frank, what's going on? I've lost everything. So we practiced this thing and everything was perfect. Okay, you think it's going to work now?
Yes, it is. You click the button, don't click up here.
Well, you know what I'm going to do? I'm going to just use this.
Okay.
Is it for what's going forward?
This
one?
Yeah.
No, it's not.
Yeah, that one. Yeah.
Okay. So there are a lot of research questions with this virus that are still remain very unsolved, open issues for people to do research, young people on, including even therapy for the aggressive leukemia. Very, very poor therapy for it. But if you can read the slide, I don't have to go through it, but there are a lot of fascinating questions. One of them, for example, it obviously leads to immune impairments, but it completely differs from HIV, which leads to more kind of fungal infections and viral infections. This is purely bacterial bronchiectasis. How? Why does it occur with this subtype C? How is it causing modest immune impairment overall? You know, is it too late to use antiviral therapy like we do with HIV for early stage HTLV? We don't know. And vaccine, this one, if you have a chance for a vaccine against a retrovirus, this would be the virus. Anybody doing it? Maybe in Japan. Hardly any funding for it in the United States. That's a problem. Okay, so let me see, where did I go over here again? So discoveries of these viruses, 1 and 2 of HTLV, in 1979 to 1982 made it possible, in my view, to both technically and conceptually make the discoveries of HIV in the 83-84 period. What do we know about AIDS 1981 and 1982? Before the virus was discovered, what did you think we knew about it then? Now, there were some people who were around at that time. We knew from clinicians, and this is a scientific contribution, that CD4 positive T cells were markedly reduced, thus suggesting to me, of course, and to others, I hope, that CD4 T cells might harbor an infectious agent. The mode of transmission was very likely to be by blood, sex, and mother to newborns, by US epidemiologists telling us that drug addicts had it more, blood transfusion, hemophiliacs, sexual promiscuity, and the babies born to mothers that had AIDS. We also knew that it was prevalent in Haiti and among gay men at the time. And we knew of suggestions of an African origin, and we knew that the disease was over a long period. Every one of these things suggested to myself anyway, that this was going to be caused by a retrovirus. Max Essex at Harvard thought the same thing, and we began communicating with each other at this time. The ideas for the cause of AIDS began in 1982, that at least putting them out there. The notable theories included non-infectious, which always seemed to be a little bit weird. Poppers, for example, but how could that explain babies, and how could it explain blood transfused people and all the hemophiliacs, et cetera? And autoimmunity, that was even further removed from the possibility. think about it, and you'll understand why I don't think those were very good theories. Okay, then there were good ones that were infectious, and the one in orange turned out to be the productive one. We proposed it with Max Essex at Harvard in the 82 period in lectures and in some science magazines, but not real formal publications. While our papers were in press, some of our neighbors at NIH had a paper in the New England Journal that was retracted that a fungus was the cause of AIDS. That turned out to be a contaminant. And then there was the ridiculous that there's no AIDS, or there is AIDS, but there's HIV doesn't cause it, or there's really no HIV, et cetera, et cetera. And then, yes, it does cause AIDS, but it was deliberately created by the United States government, of course. All of those were some of the things that we were having to go through at that time. I wanted to bring out that finding the virus was one thing, but showing causation was quite different. I think AIDS presented unique challenges unlike past viral epidemics, or the recent SARS-1, MERS, or the current pandemic of SARS-2. What was unique about it was its clinical latency. Doctors don't ask people what they did 5 years ago, or 10, or more. You ask what they did last week, last month. There were multiple other microbial infections. How can you say which is causative? There was difficulty in reproducing the virus isolation because some AIDS patients had hardly any T cells left in their blood. We were very much helped by certain clinicians, particularly James Oleski in New Jersey, Redfield and his colleagues at Walter Reed Army Hospital, Jim Hoxie at the University of Pennsylvania, Bart Haynes at Duke, Jerry Groupman at Harvard, Mark Kaplan at Cornell, and Sam Broder and Bob Yarshorn at the National Cancer Institute. Not just in supplying samples, but in discussions. This set us back. I've never published this. Well, Johns Hopkins pathology professor published it in the cover of his journal, but I never formally published this. But it set us back approximately a half a year. One of our earliest virus isolates of HIV was from a young man who was having a honeymoon holiday in Haiti. And like many people who go to Haiti, he gets in a car accident. And unfortunately for him, he required blood transfusion. He developed AIDS pretty rapidly. We were able to culture his cells permanently. This was the first time. And we were succeeding in showing a retrovirus was really present in high amounts. Previously, we were getting positive results followed by many negatives. We'd get another positive particle containing reverse transcriptase that wasn't exactly seemingly the same as HTLV1 and on and on and on, but it was really close to HTLV1 and HTLV2, causing us confusion. Was it in the HTLV family? It must be so, etc. And then this comes along, and this just built right into my bias. Namely, it is a retrovirus cause of AIDS. It is killing these cells, yet the cells can have a good proliferative rate because it's got the proliferative angle of HTLV1 in it. Of course, we were thinking it's all one virus. It is known that two retroviruses don't usually infect the same cell. There's something called interference mechanisms. But after some time, we realized we could culture out HTLV1 and separate it from HIV. We didn't know HIV, the new retrovirus. And so the light came. My God, this is a doubly infected person. He's in Haiti, which is an endemic area of HTLV1. He got a blood transfusion, he got HTLV1, but he also got this new retrovirus that must be the cause of AIDS. My laboratory at the time was Marjorie Guroff, and she was doing seroepidemiology, and she came to see me about the same period of time, a little later, and said, you know, those samples that I'm seeing antibodies positive for HTLV1, I'm convinced are HTLV1. It's a double infection. There's something else still there. And you see what else is there and what the confusion was about. Then things moved rapidly. So we now could understand what was going on. And when we first published, we published 4 papers in Science and 1 in Lancet at the same time in the spring of 1984. We reported on 48 isolates from 48 different patients with AIDS. Our first report in 84, and then 105 isolates in the early winter of 1985 in the proceedings of the US National Academy of Science. Those isolates, as well as some additional isolates from Paris, were still nonetheless insufficient, in my mind, to conclude that HIV was the cause of AIDS. Because in science, we know, at least in our country, that it's necessary for science to progress that you need to be verified. To be verified by virus isolation was going to be very difficult because tissue specimens were limited and not even allowed in some institutions because of fear. Because T-cell culture technology was not widely available in virology labs at the time. And because AIDS had to be clinically recognized, and by that time the patients had very few T-cells to culture, making virus isolation tricky. The consequence, very, very few groups were involved in attempting to isolate virus from those T-cells. The fall of '83 came a key advance, the capacity to continuously produce the virus in a cell line of several of our isolates of HIV. And it made a lot of things possible, like the blood test, like drug screening, like detailed molecular analysis of HIV genes and proteins and so on. And my key colleague in this work was Dr. Mika Popovic, who came as a visiting scientist from what is today the Czech Republic. But no, I think it's Slovakia. Yeah, and stayed. He's still at IHV with us, at least part-time, right today. So what I want to say is that if we go back to the cells with the double infection that caused us the confusion, it also was a boon for us because it led to the realization that an immortalized CD4 positive T-cell line, that is a T-cell culture that can grow forever, like a leukemic one, could be the source for producing massive amounts of this virus. Thus, on that day, on that period, we knew something definitive. We knew that we may not yet know the cause of AIDS definitively, but we know we soon will. And that was the case. Within a few months' time, we could mass produce the virus, make the blood test, do all kinds of stuff, and be able to link this virus to the cause of AIDS. Why? Because you can do thousands and thousands of sera. You can't do thousands of cultures. So the blood test was able to verify etiology because it's safe, simple, sensitive, accurate, inexpensive, and rapid. Verification was almost instantaneously. It enabled surveys of thousands of sera globally. Sorry, how do I get rid of that? I don't know. Oh, Jesus, I gotta throw this mouse away. Can you click that off? Sorry, folks, I'm losing time and that's not in my interest, but it's, I did something that led to okay, he's fixed it. Our IT guy, Frank Pfeiffer, who I have to acknowledge, just fixed it. Okay. The blood test for many of us was a key advance. It saved the blood supply. It prevented blood transfusion infection as well as the consequent infection from recipients. It allowed the epidemic to be followed for the first time. The culture system allowed the first screening of anti-HIV drugs. And when therapy became available, we could determine who to treat, as well as block mother-to-child transmission. And critically, it also verified HIV as the cause of AIDS, because thousands of sera could be tested, as I've already said. About therapy and prevention, we have to think of it, remember that viruses are inanimate, have no metabolism. So to target a virus, the middle concept here, with drugs, is vastly different than targeting bacteria with antibiotics. It used to be thought in my early career that this was more or less impossible to achieve because of the extremely high level precision needed for therapy. But advances were coming from precision molecular biology, making those views archaic. Thus, it was essential first to understand at least some aspects of the HIV replication cycle, and so it was. The next came the definite evolution of anti-HIV therapy In animal studies in the 1970s, we blocked retrovirus leukemia by treating newborn AKR mice. These are mice that 100% will develop leukemia by 1 year of life. But if we treated the mother with a reverse transcriptase inhibitor, we could prevent the leukemia. But that's lost in the literature. And frankly speaking, I don't even know why I did those studies. We didn't have a human retrovirus at the time. We just did it. But in 1984, after the cause was demonstrated and the blood test developed, the culture system was available. Sam Broder in the National Cancer Institute at the time in Tom Waldman's group, and then he eventually became director of the National Cancer Institute. He came temporarily to our lab and worked in the culture systems we had available and wanted to work on therapy. And I had been invited to Burroughs Wellcome to come for lecturing at Duke and to visit Burroughs Wellcome. And Broder came with me. And in collaboration with Burroughs Wellcome, we showed that AZT had relative and selective activity against the HIV reverse transcriptase compared to human cellular polymerases and cell-free enzyme assays, and also markedly inhibited HIV replication in cell culture. The period is now 84 and 85. Without Broder, this would not have happened. But the first work, yes, came within our lab. But then Broder and Bob Yarshorn, still at NCI, brought it to the clinic, 85, 86. And for the first time in the history of medicine, not well appreciated, showed that a systemic viral disease could be treated. This brought many pharmaceutical companies into anti-HIV drug development. It opened up more generally the concept that virus diseases can be treated. The concept of combining a few different drugs was obviously important, but it was also obvious to do. We simply had to wait for drugs to be made, work at different levels of virus replication, or even at the same level, but by different mechanism. By 94, the stage was set for the cocktail that made HIV/AIDS a disease one can live with. There were great contributions from a pharmaceutical industry, maybe particularly Gilead and its founder, John Martin. However, the requirement for continued therapy and the presence of some comorbidities promotes a view that more has to be done, thus the cure push. I haven't followed that. I mean, I don't think that that's going to occur because I don't think it's possible to ever prove without total dissection that a person is absolutely cured of all proviruses. I think we need to push for and search for long-term therapy that is not toxic, that can be given periodically only so people will remember, and easily given. This would be the final dramatic advance, I believe. Now, vaccine against HIV. I want to say just a few points about it. It can't be entirely based on principles of classic virus vaccinology because it differs in critical ways from those we have succeeded in making vaccines. The critical major differences is the great error rate, the great variability of HIV, but more importantly, that its genes integrate in the host cell chromosomal DNA and occurs in less than a day. It soon establishes infection and provides little time for immune recall. Therefore, a protective immune response must persist. That's not before required for other vaccines. And complete block of infection, sterilizing immunity might be required, never before a requirement for a successful vaccine either. An old approach from our Institute of Human Virology and earlier in NCI was that the vaccine should make protective antibodies. The antibodies likely should target GP120 as the first thing the cell sees and initiates the infection with. The antibody must be broad, targeting conserved regions, but regions known to be functionally required. But these sites are hidden. So my colleague at the time, our colleague, Dr. Anthony DeVico, working closely with Dr. George Lewis, opened internal sites by methods which also fixed the mobile GP120, because GP120 is normally in a dynamic state and antibodies have a hard time finding sites. This allows new sites to be exposed. This was a change carried out after moving to IHV in 1996. You should note that your own Marzena Pazier carried out some of the key work on the structural biology of the molecule. Let me just spend a moment more on this. This slide shows the various stages of HIV entering the cell. The virus GP120 component binds to CD4, shown on the left side. Once it does that, the bean-shaped GP120 changes, the oval-shaped GP120 changes its shape to bean shape. shown as the third virus in going from left to right. That allows it to interact with the co-receptor molecule known as CCR5. This allows the fusion events to occur, where you see on the far side the membrane of the virus fusing with the membrane of the cell. The guts of the virus are then emptied into the cytoplasm of the cell. So now, what is the vaccine? The vaccine that was devised by Tony is GP120 bound to the domains called D1 and D2 of CD4 through a linker molecule. That becomes the vaccine itself because it opens up sites that are searching for CCR5, new epitopes, so to speak. And now you see this as it would be represented, the GP120 in red, the linker of neutral amino acids, the D1, D2 of human CD4. We also have the monkey CD4 because we're going to use monkey experiments and you don't want antibodies going against CD4. Now, let me tell you, when we used that vaccine, what the results were. This is some of the studies that Marcena were involved in. I don't have time to go through this, and I have to just quickly run to this last of these almost last of the vaccine slides. You look on the left and you'll see what happens when we have non-sterilizing protection. Both groups, vaccinated and challenge group, still have virus, as you see in the plateau in red and yellow, vaccine group and challenge group. But now if you have sterilizing protection, the vaccine group is protected. We got that in monkeys with this vaccine. Oh boy, hurrah, hurrah. But in time, the red line approaches the yellow line. That is, after about 14 challenges or about 14 or 15 weeks, we lose the protection. That has been a problem. In attempting to solve the problem of lack of durability of the antibodies caused other problems. It activated too many T cells, too much T cell activation. Activated T cells are the home of HIV. HIV replicates in activated T cells. So you're shooting yourself in the foot, and that would be a problem. So you have all these activated T cells at mucosal sites, and you tip the balance and you get infected. In other words, you could have a successful vaccine and you'd never know it because you have so many activated T cells that you lose all protection. So what do I really think? I think we need a more creative approach to an HIV vaccine. I don't think any of the currently available vaccines, including our own approach, are going to be an answer. And I think there should be more funding for young people doing creative science in this field, and not everybody working on neutralizing antibodies. It's a strong opinion, one I could spend a half hour with. We don't have time. In my remaining minutes, I'd like to tell you what I think about SARS-2 in COVID-19 disease. You and I know that there were 4 great pandemics in the past 100 years. Flu, 1918, polio, the 50s, HIV, the late 70s, early 80s, SARS, 2019, right? About a generation in between each. So when you hear somebody say, there's another great pandemic, of course, coming, there's a, it's not if, it's when, it'll be maybe next year or 2 years. This, by history, is nonsense. It averages about after a generation or so, just time enough not to remember. I mean, many people say we'll never forget this pandemic. My guess is we will. We're living to see the forgetting of AIDS already and HIV. People don't even list it on some of the great pandemics in their talks anymore. We don't remember. We didn't remember the flu of 1889 in 1918. Read Oshansky's book on polio. We didn't remember the flu of 1918 when polio came. I was there with HIV. I can sure tell you, we didn't remember polio in the 70s. We did in the 60s, not in the 70s. My conclusion, I joke in a way, my tongue is in my cheek, but maybe it's true. Humans have a memory of approximately 1.5 generations at max. It's interesting that all these viruses are RNA viruses and that all these pandemics were caused by RNA viruses. They also cause multiple epidemics: Ebola, SARS-1, MERS, Zika, dengue, chikungunya, West Nile, flu repeatedly. They're all RNA viruses. They all have high mutability as well, except and they all have high replication, except What did I say there? Yeah, except HIV is not so high. Easy transmission, rather. Geez, it's written wrong. I'm sorry. They all have these characteristics of high mutability and high replication and easy transmission, except HIV, not easy transmission, as you all know well. When you think of the origin of a pandemic, think of one word: change. It's always there. So you might say, I used as an example today in talking to some others, Lyme disease, deforestation, the deer and the tick, right? Comes in your backyard. Legionnaire's disease, refrigeration. Many epidemics in the times of antiquities. Romans coming to a new port in the Eastern Mediterranean. They developed new epidemics, both directions. Europeans coming to the New World. New epidemics, particularly for the Native Americans, but also maybe syphilis returning to Europe. What other new things can we think of? I think that we should note that polio, I can't think of, the Great Flu epidemic, World War I, and the concentration of soldiers in really crowded conditions. What can we say about SARS-2? I don't know. But I think if we want to know the origin of SARS-2, we should start thinking of what happened in Wuhan since the time of SARS-1 and a few years before it. You'd probably start a few years before that time, because that's when these coronaviruses really blossom forth. SARS-1, MERS, SARS-2. In one case, polio, I couldn't think of what was new, but then I realized that the virus changed. It became neurotropic. So in all, we can argue for the one-word change. How I think of a virus as a possible disease cause, or thoughts on what a virus may cause with a virus in hand. What would be the disease? Yeah, both ways you can look at it. Don't look at the slide, but if you can just hear my words, it might be useful to someone. I don't know, maybe particularly a young person. But when you think about AIDS came along, this was kind of not obvious. I don't want to say obvious. It wasn't obvious, but if the disease, if you know the type of cell, you're betting that it's CD4. So you're thinking the virus must be CD4 tropic. Okay, that's one characteristic. With a long period of disease, that also fits a retrovirus, as does CD4 tropism in animal, many. And in man, we already know that all we have were CD4 tropic human retroviruses. And then you think of what kind of immune response there is to it. But before that, how is it likely to be transmitted? What are the risk factors? Blood, sex, mother to child? What Jim Kern at CDC was telling us? Well, that fit perfectly what we knew about many animal retroviruses, and certainly about HTLV1. So I just wanted to say that's how to approach. And with the disease, I think of as virus, I go the other direction, just reverse the order. Now I want to summarize and be able to come to the close of the talk. How have we done? You know, you always hear that. The diagnostics and advice was slow. Leadership wasn't great. I think there wasn't a great deal of great government leadership. But I don't believe in blaming CDC for the lack of the blood test. The blood test for HIV came from NIH, not CDC. Who said it was only the responsibility of Dr. Redfield and CDC? We all failed. Our institute did nothing for developing a blood test for this. We didn't even try. We thought it was already handled, it was going to be pretty simple. We didn't pay any attention. So I think it was a failure of medical science, period. Okay, therapy. Early antiviral specific therapy was again disappointing globally. But I'm not sure if that's due to science. Is it due to our fault? This time I think it's due to the nature of the beast. The viremia, the presence of the virus, its real role is early and fast. It's very hard to get a pill suddenly, available fast right there and then. Injectables will be more readily available. This is a difficult nut to crack, the antiviral therapy. We will see that the anti-inflammatory things were not great from the government, the hydroxychloroquine push, the plasma infusions, and even remdesivir. Dexamethasone was working, we heard from Italy. That's been helpful. But I promise you that true advances are soon coming. One of them, a major one, from a man that was at our institute, Yang Liu and his wife Pan Zheng, now at their company, OncoImmune, sponsored by Merck. Their drug, which is a CD26 FC chimeric, is having rather amazing results in a big study nationwide and will be soon submitted to a clinical journal for publication. It's going to be a major advance. But I want to point out that I'm not keen on having government necessarily leading the virology in the future. It'll probably have to be the case, I suppose. But you have to ask yourself, what government? Did we agree with Boris Johnson or with Sweden? Or should it be the Japanese government or the Chinese government? What government? I don't think it should be. I think we need the government for funding. We need the government for oversight. And we certainly need the government for making positions. But in my view, we need international virology leadership for the science part of this. And we need them linked and, in ways, the foundations, in ways that we haven't done before. And I'll come back to that as I close in a minute or two. But let's hear a minute about the vaccines. So far, clearly so good. Like many acute viruses, this has not been so difficult. Pretty easy virus. Once the key information came from the Chinese publication in January 10th of the genomic sequence, you could buy the sequence. Pretty easy to get the mRNA, for example, or get the protein. However, the technical advance was considerable in protecting that RNA by lipid chemistry and by using the principles of nanoparticle technology, etc., and getting membrane fusion. This has been a serious advance. So those vaccines are impressive. Any limitation? My view is that the so-called second generation are really the same. The only difference is the method of delivery. Virtually, the eggs are in the basket of the spike antibodies. T-cell-based effective vaccines might be improved with additional SARS-2 proteins. I think that should be given serious thought. But once again, to conclude on that, so far so good. I'd like to close by considering innate immunity. Will specifically stimulating innate immunity be helpful? I published a couple of papers on this, and I am not an expert on innate immunity. I come from the outside, and never have I seen a field so important, so very important, so underappreciated, and so underfunded as what this might do. We were proposing a year ago, February, to go forward with live attenuated vaccines, non-specific vaccines that cross and stimulate innate immunity, which gives temporary, now known to be as long as several months, partial protection, fairly good protection, sometimes better than the specific vaccines that have been shown, as has been shown with OPV in Russia against influenza in the 1970s. I took measles, mumps, rubella, as did my wife, as did all the leadership of the Global Virus Network, this international organization of 61 centers of excellence throughout the world. We were all taking MMR in the absence of the available vaccine. Nelson Michael, your Colonel Nelson Michael, who's a friend of mine and I was in close contact with, also understands this concept and believes in this concept. I believe that if we hit this in February when we went forward for funding at NIH, we would have saved a heck of a lot of lives and dented this epidemic, this pandemic. I believe it firmly. There is a reason why people might not want to get excited by it. They don't understand it enough. They don't believe it enough. They don't appreciate it enough. But more importantly and substantively, they had to deal with the specific vaccines, and causing confusion in developing the specific vaccines was not something anyone was looking forward to. So little attention was paid to this. I believe stimulating innate immunity to be tremendously important for the United States military. I believe, I used as an example today, a new epidemic breaks out somewhere in the Middle East and you have to go to that place. If you had in your, no vaccine, no therapy, but you had in your pocket oral polio, safe, simple, cheap, 15 cents a pill, available readily, or if you don't like oral polio, I can go to my drugstore and get measles, mumps, rubella. They work. Believe me, they work. I will recommend you read an article published by a dozen of us that should come out in the proceedings of the U.S. National Academy of Sciences. I'm hopeful in the near future. And that article will summarize pretty much all the concepts of it and why it's probably absolutely essential for many nations in the developing world to take advantage of. So preparing for the role, and I think this is, I believe, my last slide. Government role, in my mind, should be funding, oversight, some key decisions, of course. But I think it's proven that different governments differ in their thinking and their approach. So we'd have to ask, what government? Is ours always the best one? Governments select a handful of advisors, but are those advisors always necessarily the optimum? Private groups, I've already said this, banding together, like the Global Virus Network with CEPI, with Gates Foundation and Gavi, working with WHO and reporting to global governments. Governments that are united in some way. With greatly improved local public health that I think has declined a little bit in our country. And consider innate immune stimulation by live attenuated vaccines. And maybe they don't have to be live. I've heard that a paper is going to be coming out very soon that inactivated influenza has had a substantial impact on COVID-19 already. And I'm submitting a paper with a group in Iran. I hope that's okay. And the Iranian publication we're just submitting now for clinical group, our group in collaboration with them, is shown a major difference if you've been vaccinated with OPV in Iran versus recently, or if you've been exposed like a mother of a baby where there's been vaccination, is protected compared to the placebo group or the control group in a very statistically significant manner. So these studies will be coming out very soon. I do thank you for your time and attention, and I particularly thank Lieutenant Colonel Myatt for his courtesy and kindness to me, and for Marzena for the same, and for giving me the extra minutes without, I know it's important to be on time in the military, and I have a little bit abused that time, but thank you.
Dr. Gallo, we have certainly enjoyed your presentation. Thank you so very much for taking the time to present to us. And we're now going to open up this portion for question and answers. I'd like to lead off with a question, and if there are others in the audience who want to present questions, they can present those through the chat line, and Major Yarnell will present those questions to Dr. Gallo.
Thank you for your comment.
Dr. Gallo, you've been at the forefront of scientific discovery for decades, and there were so many of your slides you shared with us that were a demonstration of that. What approach are you most comfortable with for future validation of viral causality of diagnosable disease?
To the evidence for the virus causes of disease. Very hard because for some viruses because you can't fulfill Koch's postulates. For HIV, we could, because there were, I don't know if you know this, but there were a number of technicians that were accidentally infected. Not in my lab, thank God, but there were, and with clone of the virus. They died pretty rapidly. Not rapidly, but within 2 years they were dead. And in a few cases, this is not well known, but I mean, but even the blood transfusions, you were fulfilling Koch's postulates in humans like straight away. So we have it with HIV all the way, right? You have an unusual, I said in a book I wrote that a large part of it is the source of common sense, but it's more complicated than that because you do need statistics, and statistics are not just common sense. But you certainly start with, does it fit your intuitive thinking? Does this kind of virus do that kind of thing? A disease like SARS, there's no argument. Nobody ever talks about showing the cause of the disease. It's obvious in your face. It's dripping from the tongue of the person who's sick, right? And the person who's well doesn't have it. It's always there and it's coming on. You don't have to be a magician to find it. But in a slow virus, it's another story. And when you speak of that, you're speaking like papilloma causing cervical cancer. Long, arduous task to get the acceptance of that. HTLV1, the viruses that are slow viruses. That's the hard ones. The acute viruses, you're never going to argue about. No one is. Ebola breaks out. You're not going to say, let's go prove Ebola causes Ebola. It's evident, right? It's right there. Acute virus infections, it's evident. So you don't need to argue about it. But when you have something, let's say you believe, as many people do, that multiple sclerosis is due to a virus. What kind of virus should it be? How are we going to prove it? With multiple sclerosis, it's called the graveyard of the virologist, because there have been many claims, no proof. So that's really a tough one you get involved in. What would I want to see? That it logically fit. It's the kind of virus that makes sense. Then I would want, number one, the close epidemiology that fit without any real strong exception. The groups at risk that are the same groups at risk for this bug, right? An animal model that you can reproduce the disease with the genes of the thing, let alone the whole virus. And if you don't get an animal model, it's not because you're wrong. Don't give up, because viruses don't infect all animals. They tend to be species specific. But could you make a humanized animal model, for example? Could you make a transgenic using the genes? Would it fit? What happens when you add the virus to what you think is the target cell in culture? What does it do? Does that make intuitive sense? HTLV1 transforms the T cell. It immortalizes it, makes it grow forever, and those cells look abnormal, and it makes them abnormal. Well, that's interesting. And if you put them in an animal, they produce the same kind of malignancy when you inject the animal. That's fitting. And the epidemiology is fitting. I mean, which one would you want? In the case of HTLV1, you had this wonderful clonal angle. You don't have that with papillomavirus. You don't have the molecular argument as well, anywhere near as well. And with papillomavirus, sometimes you need cofactors. But I think eventually Harrelshausen did really prove it as much as you could get. And that was molecular data that was linkage. But there's still more you'd like to see. We don't have great animal model. We don't have the in vitro effect. Or, we don't have a lot of the stuff we have for HIV or HTLV1. But it's a much more important in numbers disease. So that's Harrelshausen got the Nobel Prize for it. I mean, it's a very important contribution, but you could say it doesn't come close to fulfilling Koch's postulates, but and I know that is the cost. Now that you have a vaccine and you'll be able to prevent it, well, then I think the final nail is in the coffin. So it's variable. It depends on that. It depends on the disease a lot. Okay, for a virus. But I hope this roundabout answers somewhat.
Oh, no, absolutely it does. You were spot on with linkage right there. So thank you. And I believe Dr. Quarterpeter has a question or two for you.
Yes, Dr. Gallup. Thank you again for a very interesting lecture. And it was really fascinating to see just the idea of how the different building blocks that were required before the discovery of HIV that led to the discovery. I do have a quick question, I think. Because you talked a little bit about the challenge of developing an HIV vaccine. And then also a little bit about the new approaches for the COVID vaccines. I'm just wondering, do you see any potential utilization of sort of these newer approaches like the mRNA, nanotechnology, and the lipid envelope to try to get something into the cells efficiently, such that, could this be used as an approach for HIV with some other method of developing, producing proteins in the body to fight HIV? Do you see any possibility for that as a future technology to assess?
Mark, you couldn't have asked me a more dangerous but interesting question. Dangerous because I am right sometimes, but I'm wrong sometimes. And I know what I think. But you did ask me what I think. And I think it's going to be a waste of time. And I don't think remember, it's an opinion. Please, it's not a statement that this is what's going to happen. It's also dangerous for me because one of my best postdoctorals was Paolo Lusso. And Paolo Lusso is working at NIH right now. And Dr. Fauci gave him a lot of money to do exactly that with messenger RNA, right? To make an HIV vaccine with the messenger RNA of the envelope of HIV. And so Paulo's doing great. And anything I say negative about it wouldn't be very nice. Because the work will be good, he'll do it well. But what I say not nice is, do I think it will work? No, I don't think it will work. And would I work on that pathway? Would I advise somebody to work on that pathway? No, and I'd probably make some fun about it too, because that's my nature. But I can be very wrong and then have to say I was very wrong. And I could give examples where that's happened, so I couldn't be very wrong, but no, I don't think any of them are going to do any good for HIV. I think the HIV approach is going to be very different and very conceptually different, and that nobody is thinking about it right now. Certainly not with messenger RNA. But, in a year or two, maybe I'll be saying, oh, well, where's the crow? And cook it well, because I don't like raw crow.
Thanks a lot.
Now, the next question I see came up on the chat. It said, I heard you can't even spell Marzena's last name. That's not true.
Dr. Gallo, but I think you have one more interesting question. I think
Sure.
we have one question in the chat.
I'll take as many as you want.
Angela, do you see the question?
Yes, Dr. Gallo, the question, and it seems like it may require a lengthy answer, but potentially you could point this individual to some resources. They'd like some additional information on the Iranian trial of OPV, and specifically, how did the collaboration with Iran come about?
Wow. It was through the United States military. No, that's not true. That's not even fun. No, how did it come about? A young man works in our institute who's very sharp, who was the one I referred to, Mosajati, with Anthony Amoroso earlier in the day about thinking of the temperature zone, and first published that some of the transmission could be related to temperature and humidity. He's also worked with Marzena. Marzena will tell you he's a sharp guy, working on monoclonal antibodies to the HIV envelope. He knows a young man who would like to come to the United States badly for working in virology. That young man, extra sharp, but his father is a virologist, infectious disease guy, who's been writing to me on and off for some years for collaboration on this or that, who is sharp as a tack, and I've been impressed by. And so he wrote to me when I published a paper in Science and gave some interviews about innate immune stimulation, along with my colleague at FDA, Konstantin Chumakov, the son of the Chumakovs in Russia, who made the first demonstration of OPV working against flu. The wife made it. The husband is a famous virologist when he was alive. And his mother, the wife of Chumakov Sr., made the observation when she examined the data that they weren't getting flu when they had OPV vaccination, and it was working better than the flu vaccine. But remember, it's transient. It has to be hit at the right time. So this man, first name is Farouk, wrote to me. It started that way. And he wanted to test the hypothesis in Iran. And he works in the petroleum industry, and he has access to, they have access to a lot of people where they can do studies of many thousands of people. I had the same come for me from Mexico, from a man who is the second richest man in Mexico and owns retail stores and has 201,000 employees. But that study was interfered with by a couple of people at WHO by not being saying they're not sure about this, that, or the other. And the public health people there got a little nervous about it, so never did the study. But the petroleum industry there were simply collecting data. And we worked together, analyzing and collecting that data with my colleague, Shaim Kodalil, the director of our clinical division, who used to be for 14 years at NIH. And also with Mo Sajadi in our institute, who comes from Iran. And also with Chumakov has been involved in this analysis, who's at the US FDA. But he's also a GVN center director, because his virology is so good in polio, for example, and because of his lineage, it comes from great virologists. And we collaborated together with them and giving some input and advice back and forth and getting the data. I was sent the paper only last week, so you're hearing something that's totally unknown. And so I have the paper here in my desk. And this fellow wanted me to sign off to be the signature for sending it for publication. He didn't need, he's very, very modest fellow, but he refused to do it without him as the lead person and as the lead author. And so we're doing it together as a US-Iranian collaboration of very good souls in Iran and good clinical infectious disease people. And I'm hoping someday we'll have more of it, if we could ever merge the gap. I hope that answers the question, whoever asked it. Boy, you guys asked me controversial questions. I thought this was a conservative group.
Well, we certainly have enjoyed the time that you've spent with us, Dr. Gallo. And at this stage of our Packard lecture presentation, where we're nearing the end, we're almost done. We're taking advantage of the fact that this is our very first virtual David Packard lecture. And we are going to replicate the presentation of the memento that Dr. Margina Pazgier and I presented to you last week. So for our audience, at this stage, we'd like to draw your attention to a small token of appreciation to Dr. Gallo for today's participation in the David Packard lecture. The slide before showed a memento that was approved by the university president for us to present to Dr. Gallo. Last week, Dr. Pazgier and I met with Dr. Gallo at his home to present the memento to him on behalf of President Thomas, the Faculty Senate, the Faculty Assembly. And this particular slide that you're looking at shows our presentation of the memento to Dr. Gallo in his home. The next slide shows the hospitality that Dr. Gallo offered to me and Dr. Pazgier in their home. Dr. Gallo and his wife, Mary Jane Gallo, were extremely gracious. They offered us the best of hospitality. And it was just that measure of hospitality, along with Dr. Gallo's good humor and very personable personality, combined with the focused work of the Packard Lecture Working Group, that made this entire Packard Lecture presentation a success today. So before we end this day's event, I would like to recognize all of the working group members and others who supported the Packard Lecture event for today. Dr. Pazgier, who's been mentioned before, Major Angela Yarnell, Colonel Robert Shee, Dr. Patrick Richard, Dr. Jennifer Troutman, Dr. Jose Ortegawa, Major Jose Rodriguez, Dr. Sarah Contente, and Miss Sharon Holland. Without them, this would not have been a success. We are very grateful for your participation in the Packard Lecture Days events. Ladies and gentlemen, we want to thank you for participating in the 2021 David Packard Lecture. This now concludes today's Packard Lecture events. Have a good day.
Thank you.
Thank you, Dr. Gallo.
39 questions on chat. If you send my email to them, I'd be glad to answer them to the best of my ability. Could you hear me okay?
Yeah, of course. It was
Yes.
great. Thank you so much.
Okay. Take care, everyone.
Thank you. This was